Chapter 1 Biology Flashcards. What's light microscope's main feature? How does a TEM microscope work? How does a SEM microscope work? How does a LSCM microscope work? Why are electrons better than light? To look at organisms. TEM, electrons pass through the material. SEM, electrons are bounced off the surface of the material. LSCM, sample stained then scanned with a laser beam and scanned across different depths. Electrons have a shorter wavelength so a higher resolution is possible. What is each type of microscope used to observe? Light microscope. Whole cells and tissues. TEM sees organelles. SEM sees cell surfaces. LSCM sees an object at a certain depth within a cell. What is in the nucleus? What is DNA's 2D forms in the nucleus? Nuclear pore, nuclear envelope, nucleolus and chromatin. During cell division it's tight to make more chromosomes and other times it's loose and called chromatin. What is the endoplasmic reticulum, the smooth ER, the rough ER and the Golgi apparatus? Cell's transport system, processes chemicals then exported outside the gas-folded internal membranes. The smooth ER is continuous with the RER but less ribosomes. Makes lipids, phospholipids contains enzymes and detoxifies. The rough ER has ribosomes that makes proteins, transport and secretion taken to Golgi apparatus that are sacs with cis-trans sections. It modifies proteins and lipids. What does ribosomes, mitochondria and the chloroplasts do? Ribosomes are in the nucleolus. Manufactures proteins made of two subunits. Forms are when attaching to a messenger, synthesis 80s. Mitochondria is a membrane-bound organelle do aerobic respiration double membrane contain own DNA and 70s ribosomes. Chloroplasts are membrane-bound plant cells absorb light energy. What does lysosomes, plasma membrane and the cell wall do? Lysosomes eat cellular material, contain enzymes and breaks down proteins. Plasma membrane is a barrier gateway. Cell wall prevents bursting. What is the order of organelles that are involved in the production secretion of proteins? What are the functions of the cytoskeleton? What are the differences between eukaryotes and prokaryotes? The nucleolus, the nucleus, ribosomes, RER, Golgi body, plasma membrane. Cellular movement, intracellular movement, strengthening and support. Prokaryotes don't have a nucleus. Eukaryotes' DNA is linear in nucleus. Prokaryotes is in the cytoplasm and circular. Eukaryotes has membrane-bound organelles, prokaryotes don't. Eukaryotes has 80s ribosomes and prokaryotes has 70s ribosomes. What does a bacteria cell have? What is a protoplast? A protoplast is the part of a cell, bacteria, plants, that's inside the cell wall.